Hello to whoever is viewing our video tutorial about genome analysis of complex disease, including our example Parkinson's disease. We are a group of 10 third-year students at the Faculty of Biotechnology and MSA University. This tutorial was done under the supervision of Dr. Osama Saad and Mrs. Nuran Yahya. We hope you enjoy it. Genome analysis for complex disease is a biotechnological application that is concerned with analyzing the molecular mechanisms of the complex diseases using genetics and bioinformatics tools. This can be achieved through sequencing, mapping, variant calling, and scientific discovery to develop a cure or drug. One might ask, what makes a disease a complex one? Well, to answer your question, we'll be taking a closer look at Parkinson's disease, which is considered a complex disease as it results from a complex interaction of environmental and genetic factors. Most cases are classified as sporadic and occur in people with no apparent history of the disorder in their family. Parkinson's disease was first identified as a shaking palsy by Dr. James Parkinson in 1817. It's a long-term progressive neurodegenerative disorder with both motor and non-motor symptoms. Surprisingly, its late onset form is the most common type of Parkinson's disease, which becomes apparent at people at the age of 50. Symptoms of Parkinson's disease include stiffness of the limbs and torso, slow movement, and postural instability. Unfortunately, these symptoms worsen slowly over time. It also affects emotions and the ability to think clearly, where affected individuals develop mental illnesses such as depression and hallucinations. Moreover, people with Parkinson's disease have a risk of developing dementia, which is a decline in intellectual functions as judgment and memory. Mounting evidence suggests that Parkinson's disease is forming heterogeneous subspecies. They have an impact on the diagnosis and the expected treatment response, and are divided into three groups: mild motor predominant, intermediate, and diffuse malignant. As you can see, there is a long list of genes known to contribute in the onset of Parkinson's, and there may be many more yet to be discovered. But it must be noted that the majority of the contributing gene mutations are associated with familial cases of Parkinson's rather than sporadic. It is still not understood if the genetic changes cause the disease or has a role of developing the disorder. Parkinson's disease symptoms occur if the nerve cell dies or is damaged. Dopamine transmits signals to the brain that results in correct physical movements. When the cells responsible for dopamine-producing neurons are disturbed, the communication between muscles and brain is weakened. Therefore, the brain is unable to control the movement of muscles. Gene mutations result in disruption in the cell mechanism, which breaks unwanted proteins in the producing neurons of dopamine. This results in accumulation of undegraded proteins, leading to the impairment or death of these cells. Protein deposits called Lewy bodies appear in dead dopamine-producing neurons. If these protein deposits are not present, it is referred to as Parkinsonism. However, it is still not clear if they have a role in killing the nerve cells or if they are a part of the cell's response to the disease. Previously, an organism's whole genome or transcriptome was divided into millions of small fragments and sequenced separately, which did provide massive volumes of data. Next-generation sequencing, or NGS, provides a way of sequencing thousands of DNA fragments, and it has a lot of techniques that vary greatly in reading length, production, and quality of the data, as well as the used bioinformatics tools to process and interpret the data. This data reduces the time and cost that was usually taken using the old sequencing techniques. Using NGS, the detection of disease-associated mutations and genetic modifications has been significant. It provides researchers with the necessary resources to examine massive panels of genes for each organism. Next-generation sequencing may be used to uncover genes linked to Parkinson's disease by determining the effect these genes have on the disease. In order to understand Parkinson's disease and its genetics, the huge amount of data produced from NGS in the form of DNA sequences are analyzed through the comparison with the genomes of healthy individuals, in order to identify the genes responsible for the onset of Parkinson's disease. The first step of genome analysis using next-generation sequencing starts by the extraction of total genomic DNA, the separation of genomic specimens into a group of small fragments, the attachment of adapters to the center of said fragments, then the multiplication of these fragments, followed by sequencing using PCR with special probes, and the filtration of agarose gel once the library is constructed, and lastly, the evaluation of the library's quantity and efficiency by quantitative PCR. The second step is sequence reaction, which includes the transformation of the library to single-stranded DNA, followed by the separation of said single-stranded DNA molecules to be sequenced, and after that the multiplication of single-stranded DNA molecules to be identified with an appropriate signal. Last but not least, data analysis, which includes the assembly of sequences, followed by the comparison of the target genome to a reference genome, and lastly, the sequence mapping using bioinformatics software. The story began when Watson and Crick identified DNA double helix structure in 1953, for which they won the Nobel Prize in 1962. After that, scientists all over the world were eager to understand more about DNA, especially the human DNA sequence. Consequently, various sequencing techniques emerged for the sequencing of DNA, including Sanger sequencing, also known as dideoxy chain termination method, and chemical degradation method created by Maxim and Gilbert around the same time in 1977. In the beginning of the new millennium, the first next-generation sequencing report was created. A year later, the first human genome draft was published, and in 2005, the 454GS20 was released, which was the first NGS instrument that mirrored Sanger sequencing. Furthermore, 2006 witnessed the start of the 1000 Genome Project, which sequenced the genomes of 1,000 anonymous participants to study human genetic variation. The introduction of NGS technologies into the clinic would enhance medical care, but it would also raise major legal concerns around patient genetic identity ownership and confidentiality. Ethical considerations also include decisions to hide a person's identity from publicly available de-identified results.